Hello, everyone. Welcome. So, good evening, Europe. Good morning, America, and good afternoon, Europe. Sorry, good evening, Asia, and good morning, America. Welcome to the RNA collaborative seminars. So, just a reminder before we start. So, if you have any question, please write them in the quick question and answer section, and I will read them by the end of each talk. Okay. So we will start with our first speaker, Carine Tisné, who is group leader at IBPC, Institute of Physico-Chemical Biology in Paris. Her lab is focusing on the biogenesis, architecture, and interaction of RNAs, and she's mostly using structural biology and biochemical techniques to address these topics. She will be presenting recent work from her team dealing with the structural basis of human mitochondrial tRNA maturation. So, Carine, the screen is yours.
Okay, do you see my presentation?
Not yet.
Oh. Okay. Share it again. Is it okay?
Yes, it is, yes.
Okay. So thank you, Marc, for the presentation. Hi, everybody. It's a pleasure today to present recent data that we obtained on human mitochondrial tRNA maturation. So as you all know, RNA are highly co- or post-transcriptionally modified, and they are chemically modified at nucleobases or at the ribose 2' OH by modification enzyme, and this will expand the chemical and functional diversity of RNA. And this modification has long been regarded as molecular decoration because we didn't really know what could be their role, and also because most genes encoding RNA modification are non-essential, and when strands were depleted from this gene, we observe no obvious phenotype in condition of optimal growth. But this situation has changed during the last, let's say, 10, 15 years with the discovery of modification on messenger RNA, which really boosted this field of research on RNA modification, with also the discovery of demodification enzyme that will erase some modification, and with a lot of evidence that modification can regulate gene expression. RNA methyltransferases have also been highlighted in numerous clinical studies and disease models, and also significant technical advances have been made in the field of RNA sequencing, mass spectrometry, and structural biology. So nucleoside modification are found in all types of RNA, but it is in transfer RNA, as you can see on these schemes in yellow, that we found the largest diversity and the greatest number of RNA modification. And this modification will impact base pairing, so structure and stability of the RNA recognition by proteins, but also interaction with other RNA, and also interaction with small molecules like antibiotics, for instance. So this modification will globally change all the physico-chemical properties of RNA. And in my team, for today, I will focus my presentation on the M1A modification, which consists in the introduction of methyl groups at the nitrogen 1 of an adenosine. So this modification is found in all the different types of RNA that are represented here. We found it with high abundance in transfer RNA. Few have been found in ribosomal RNA, and it is found at low levels in messenger RNA, because when you have this modification, you cannot make Watson-Crick base pair, so the translation is stopped. So the M1A modification is involved in tRNA folding and stability, and at two positions in tRNA, at position 9 in human mitochondrial tRNA and at position 58 in human cytoplasmic tRNA, this modification have been linked to human disease. And in my group, so we study methyltransferase responsible for the catalysis of this modification in transfer RNA, and we how they recognize and bind their RNA targets, and how they catalyze the reaction of methylation. So this modification is found in tRNA, depending on the tRNA and depending of the organism, at position 9, 14, 16, 22, 58, and 57. And most of the enzymes responsible for the introduction of this modification have been described. And we found this modification with a major at position 58 in all domains of life. So we on other groups over the years have structurally characterized the enzyme responsible for the introduction of this modification. So here for the modification at position 58, here for the modification 22, and here for the position 9. And what is interesting, what I want to show you here is that even if they catalyze the same reaction, they do not use the same strategy to bind and to recognize the substrate tRNA. And here, for instance, you have trimai, that is the bacterial form of the enzyme that introduced the modification at position 58. So trimai is organized as a tetramer, trimk is organized as a monomer, and here trim-T10C need a partner to be active. And the second point is that I have indicated here in cyan additional domain of these proteins that are needed to bind the tRNA in addition to the catalytic domain. And they use different additional domain to recognize tRNA, and the structure of this domain is not shared between these three different proteins. So for today, my talk will be focused on this protein, trim-T10C, that is responsible for the introduction of the M1A9 and M1G9 in human mitochondrial tRNA. So let me introduce the biogenesis of human mitochondrial tRNA. I don't know if you see my the arrow, the pointer. Is it okay, Marc? Okay, yes. Thank you. So as you know, mitochondria generates most of the energy of the cell by oxidative phosphorylation. And in human, this capacity is dependent on the biogenesis of certain proteins that are encoded on the mitochondrial double-stranded DNA genome. And you have a scheme here of this DNA genome. And you have here in purple, blue, green, and orange the messenger RNA that encoded these 13 proteins. And the DNA genomes also contains RNA component of the mitochondrial translation. And you have here the two mitochondrial ribosomal RNA and all the 22 transfer RNA that are indicated by this little circle here. That are needed for the mitochondrial translation. And what is interesting is that after transcription, that will provide two polycistronic RNA, each one for each DNA strand. It is the processing of the transfer RNA, so the processing of the 5' end by the RNA-sp and of the 3' end by RNA-Z that will release most of the RNA contained on this polycistronic RNA. And the maturation of transfer RNA will proceed by the incorporation of post-transcriptional modification, the addition of the 3' CCA sequence here, and the amino acylation to provide fully functional tRNA for the mitochondrial translation. So you have here a picture of all the 22 human mitochondrial tRNA. And what I would like to show you is that these mitochondrial tRNA are fragile, structurally fragile. So first, because they have a low GC content, and also because they are quite weird in their secondary structure. For instance, here you have a very small loop for the D arm, and here you have the D arm that is completely absent. And this is something that is quite usual for mitochondrial tRNA. But even if they are weird in their secondary structure, they must adopt this three-dimensional structure as an H shape to be incorporated in ribosome and to perform translation. to be, this three-dimensional structure is obtained notably by interaction between the D loop and the T loop here, and this base pair here. So in this context of structural fragility, the presence of post-transcriptional modification will be even more important to stabilize this three-dimensional structure as an L shape. And in human mitochondrial tRNA, the most prevalent modification is the purine 9N1 methylation. Indeed, we found 14 M1A9 that are indicated here and 5 M1G9. 19 tRNA are modified with this modification out of the 22 tRNA. And the enzyme that is responsible for this modification is 2010C, that is a methyltransferase that belongs to the phosphate-1210 family. So that means that the catalytic domain is organized as 6 beta strands surrounded of 7 alpha helices. And the SAM binding domain is organized as a pseudoknot. So the SAM is the methyl donor, so that is, and it is indicated here. And when the methyl group here is given to the RNA, the product of the reaction is the SIH. And this enzyme is active only in the presence of this partner, SDR5C1, that belongs to the short chain dehydrogenase reductase family. And so I said you that the first step of maturation is the cleavage by RNA-sp to release the most of the RNA that are contained on the polycistronic RNA. And the RNA-sp, the endonuclease is the protein PORP, and this protein is active only when the precursor tRNA are bound in this methyltransferase complex. So this complex, this RNA-sp will process the 5' end here of the precursor tRNA, and it has been shown that the pre-tRNA remains bound to the methyltransferase complex during the other step of maturation. So the cleavage, the processing here of the 3' end by ILAC2 and the 3' CCA addition. So using cryo-electron microscopy, we have been able to solve each step of this, the structure of each of these steps of maturation. So you have here the methylation of the pre-tRNA isoleucine, here the tRNA-sp, the RNA-Z, and here the complex for the 3' CCA addition. So to reconstitute the maturation complexes, we used two different precursor tRNA, the precursor, the tRNA isoleucine, because it contains a G at position 9, and this pair of tRNA, because some tRNA are produced as clusters in the DNA, in the human mitochondria. And so tRNA histidine contains an A at position 9. And we have chosen this tRNA serine because it is the one that lacks the D arm, and its maturation is special because it is the maturation of the 3' end of tRNA is that will simultaneously process the 5' end of tRNA serine. So after reconstitution, we checked that our complexes are active, are functional, and this is what you see here. So here you have the methylation test that show that this complex is able to methylate both precursor tRNA. Here you have the activity test for the cleavage by PORP of the 5' extremity and by ELAC2 of the cleavage between the two tRNA. And to reconstitute this complex and poise the endonuclease on the precursor tRNA, we have used catalytically inactive mutants. And for tRNT1, you see on the gel that tRNT1 is able to add the CCA sequence when the pre-tRNA is on the methyltransferase complex. So you have here the non-city map that we get after processing, and the global resolution that we obtain ranges between 2.7 to 3.2 angstrom. And the local resolution from the maturation enzyme is between 4.1 to 4.5 angstrom. So I will describe now a little bit more the structure that we obtain and the information that we get. So the organization of the maturation complex is more or less the same, with the methyltransferase that is here in green, that wrapped around the tRNA that adopt an L-shaped three-dimensional structure. And you see here the partner of 2010C. And here you have the structure of the RNA Z complex. So you see here LAC2 that is bound on the top of tRNA is. So the binding of tRNA is on the methyltransferase complex exposes this part of the tRNA where the maturation enzyme can bind. And here you have the junction, so the cleavage sites between the two RNA that is positioned in the active site of LAC2. So this is what you can see here. And what is interesting here is that the 3' extension, that is the tRNA SER here, is as a binding site here at the surface of LAC2. Okay. So if I go back to the structure, what is interesting to see is that the maturation enzyme here, the endonuclease, cannot bind simultaneously. So the cleavage of the 5' end and the 3' ends cannot occur simultaneously. So this imposes an order in the processing events. And there is data in the literature that showed that this processing occur first the 5' extension is cleaved and then the 3' extension. And actually, if we look in more detail the structure that we get with LAC2, and if we put inside the structure of the tRNA with the 5' extension, you see here that there is steric clashes with this helix here that is conserved in metazoan. And if we do the test, if we perform the activity test with this tRNA, you see here that LAC2 is not able to cleave the pre-tRNA either when the 5' extension is present. And it is the same for the pair of tRNA, whereas it is active when the 3' extension, the 5' extension, sorry, is already processed. And the second argument in favor in this order is that, on the contrary, PORP is able to bind both extremities, and this is what we see in the structure, the 5' cleavage side and also the 3' cleavage side. So our structural data explain why there is an order and why this order is 5' to 3' in the processing of human pre-tRNA extensions. The second thing that I would like to show you is that the maturation enzyme make contacts with the additional domain here of the methyltransferase. So you have this part here, and the PORP is the protein that make the larger contacts. We observe contacts here and here, but to a lesser extent. And if we delete the helices here that made the contacts with the PPR domain of PORP, you will see here on the activity test, as soon as we delete the two first helices here, PORP is not active anymore. So that means that this interaction is really is required to clamp PORP on the pre-tRNA and to position here the active site of PORP on the 5' cleavage site. And this interaction explains why the methyltransferase complex is required for the PORP activity. So if I summarize a little bit, 2010C, so the methyltransferase recognizes the entire L-shaped structure of the pre-tRNA. Each arm is contacted, and the methyltransferase is also required for the first step of processing. And what we propose is that the methyltransferase has a role of tRNA folding quality control, because if tRNA is not well folded and is not able to acquire this three-dimensional structure because of, for instance, of a problem of transcription, it will not enter this chain of maturation events because it will not be bound by 2010C. And this is actually the case of tRNA SER that is processed not by this chain of events because tRNA SER is not able to be bound by the methyltransferase, and its maturation occurs through the binding to the adjacent tRNA. And this is what you see here, for instance, the 5' end of tRNA SER is processed by the processing of the end of tRNA is. And to conclude and to go in more detail of the information that we get, I will show you a movie and try to summarize what we see. So you have here the methyltransferase complex with the pre-tRNA isoleucine that is bound. Here I show you a zoom on the tetramer or the partner of the methyltransferase, and that is a tetramer, and you see here a molecule of NADH that is bound in each subunit. You see here the contact with the methyltransferase on the anticodon loop. And I will show you a zoom on the active site of the methyltransferase where you see the adenosine 9 that is flipped out and stabilizing the active site, and you see here an SAH molecule. So now you have the second precursor tRNA, and I show you a zoom with the guanosine this time. That is so it is this atom that will receive the methyl group, and the methyl will be here and go here. And you see in the active site also the conserved residue that stabilize the adenosine. So you have now the binding of the PORP protein to form the RNA-SP complex. And you see here a zoom on the active site of PORP that is bound on the 5' cleavage site. So PORP with one magnesium ion, and we see only one because of the mutation, but normally two magnesium are present in the active site. I will show you now this specific interaction between the PPR domain of PORP and the methyltransferase that allow to position the active site on the 5' cleavage site here. And then you will observe the first factor exchange between PORP and ELAC2. And you see now ELAC2 that is bound on the pair of tRNA. Yes, and you see here the binding of tRNA SER at the surface of ELAC2, and you have a zoom now on the active site. So with one zinc ion, but normally it's two, and because of the mutation, we only see one, and you have here the cleavage site. And then you will see the last factor exchange between ELAC2 and tRNT1 that binds on tRNA isoleucine after processing the 3' end processing, and you see here the last nucleotide and the stacking of the first of the CDP, the first C that will be added for the CCA sequence. Okay, so with this, I would like to thank all the people who contributed to this work, and in my team it was mainly the PhD work of Vincent Meignier, our collaborator, our financial support, and I thank you for your attention.
Thank you very much, Carine. Is there any question? I don't see any question in the Q&R, so if you have any question, feel free to write your questions, please. Well, maybe I'm going to start before people write their questions. So you nicely explained to us that the methyltransferase domain is orienting the tRNA towards the active site of the different enzymes. So my question is the enzymatic activity necessary in vivo, the tRNA methyltransferase activity, or can we imagine that only the fact that it's going to orient properly the tRNA will be sufficient, and then enzymatic activity might not be so important?
The gene of 2010C can be silenced, but the cells are not really happy.
Yeah.
The thing is that we think that this modification is essential is the first modification that is introduced and is essential to stabilize the three-dimensional structure. And it is why it is also so prevalent in this tRNA. And probably some tRNA, when the enzyme cannot make this modification, the mitochondrial tRNA are really, really fragile or degraded.
Okay, thank you. So two questions now in the Q&A.
Okay.
One is about the role of the D-arm. Is the absence of the D-arm affecting the processing of the tRNAs?
The absence of the D-arm makes that the cell has invented another way to mature this tRNA, and this tRNA is matured by the maturation of adjacent tRNA. So this tRNA serine is included in a triplet of tRNA, and it is the tRNA of the maturation of the end of the two other tRNA that matured. So the methyltransferase recognize the tRNA that are adjacent, but not the tRNA serine.
Okay. Two related questions regarding the methyltransferase activity. So when is the modification occurring? Before or while the 5' 3' processing enzymes are bound, or is the 5' 3' RNA processing
We don't know.
to release the modification enzyme?
There have been a lot of kinetic experiment in the literature, but it is not really known when the methylation is occurring, and it is not sufficient to release the tRNA. Okay.
And someone is asking whether the modification takes place in the mitochondria or cytoplasm.
Mitochondria. It is the transcription is made in the mitochondria, and the tRNA are matured in the mitochondria.
Okay. And so some people are confused about mitochondrial RNAs, tRNAs. So are they synthesized into the mitochondria and then they remain into the mitochondria, or they go back and forth to the mitochondrial?
No, all the RNA that are needed for mitochondrial translation are produced in mitochondria. So the ribosomal RNA and all the 22 transfer RNA are produced in the mitochondria. Okay. Only the protein are imported into the mitochondria.
Okay, and this means that the machinery that goes to it's the enzymatic machinery that goes to the mitochondria, right?
Yes, yes.
Those are encoded by the nuclear.
Yes.
Yeah. Okay.
The only protein that are produced in the mitochondria are the 13 essential protein for the respiratory chain.
Okay. Yes, no more question. Well, I may ask one last question. So when you don't have the D-arm, does the tRNA look like a classical tRNA with an L shape? I mean, could the T-arm be longer to compensate that, the absence of the D-arm? Or?
There is a structure now of this peculiar tRNA in complex with the synthetase. And well, there is an angle quite well, but he has more or less this L-shaped conformation. So the anticodon, I think that the anticodon arm is longer. And even if he does not have the D loop, the D arm, he has a form that resembles an L shape.
Okay, thank you very much, Carine. So I don't see any. Oh, one more question, maybe. Yes, one last question. Did you generate a catalytic mutant of TRIM-T-C? And so, if the TRIM are the cells sick as well as the KO cells? Do you know that, or maybe?
I don't know. We did not generate a mutant.
Okay, so. Okay, so there's one more question, so maybe we can shift to the next speaker, and then you can answer the question in the Q&A.
In the chat. Okay.
Thanks a lot. And
Thank you.
now let's move to the next speaker, who is Sébastien Campagne. So Sébastien is group leader at IECB in Bordeaux. So IECB stands for European Institute of Chemistry and Biology. He is using a multidisciplinary approach combining structural biology with biochemistry, biophysics, and cell biology to uncover the underlying mechanisms responsible for regulation of RNA splicing. And he will talk about small molecule splicing modifiers, a historical perspective. So, Sébastien, please.
Thank you, Marc. So welcome, everyone. So the talk today, as you say, is entitled Small Molecule Splicing Modifier, an Historical Perspective, because it's already 10 years that the first small molecule splicing modifier has been discovered. And from this time, we understand now its mode of action, and we start also to design some new challenges. So, as you all know, the eukaryotic genes, they are transcribed as precursor messenger RNA, because they are interrupted by non-coding sequences that are called introns, and RNA splicing consists in the removal of these introns and the joining of the coding sequences that are called exons to form the messenger RNA that will be then exported into the cytoplasm, where it will be translated, degraded, or stored. So the sequence of the messenger RNA will determine the sequence of the protein, and of course, this will play on its function. And the beauty of this phenomena is that splicing can be regulated, and from a single pre-messenger RNA, one can obtain different messenger RNA, different proteins that will be associated to different functions. So alternative splicing increase the coding capacity of the genome, but also regulate gene expression. And in a cell, basically, one has to see that as an equilibrium. And when the equilibrium goes in the right direction, basically people are healthy. And when things goes wrong and non-physiological splicing pattern occur, this is often associated to human disease. So how is splicing done? So it's catalyzed by the spliceosome. It's a long reaction that need the intervention of a lot of U, SNRNP, and different associated protein. It's a stepwise assembly of the splice. So the spliceosome is assembled in a stepwise manner. So in the first phase consists of the recognition of the splice sites that will form a platform in order to recruit the catalytic core of the spliceosome, and then follow several rearrangements and the catalysis occur. So basically the intron area is released and the two exons are linked together. So nowadays we have an atomic view of this splicing cycle, since most of the intermediate structures have been solved at atomic resolution. However, the regulation of splicing is still poorly understood. And it mainly occurs during the assembly phase, during the recognition of the splice sites. And us in the lab, we are particularly interested by the recognition of the 5' splice site by U1 SNRNP. So this recognition step is basically done by a direct annealing of the 5' end of the U1 SNRNA directly on the 5' splice site that lies at the exon-intron junction. It can be modulated by several factors, including the complementarity between U1 and the 5' splice site, the formation of inhibitory secondary structure that will mask or reduce the accessibility of the 5' splice site, and finally by trans-splicing factor that will bind specific cis RNA sites at the surrounding of the splice site, and then will modulate the recruitment of the splicing machinery, or in this case, U1 SNRNP. So 5' definition is definitely a hotspot for splicing regulation, but also for splicing correction. And there are many mutations that cause disease that are falling on this 5' splice site. So you can see here a diagram from Emmanuel Verratti that was published in 2007. So where you see the frequency of mutation causing disease on the different position of the 5' splice site. So you see here at the bottom the consensus sequence of the 5' splice site. So you see that there is always a GU at the beginning of the intron on which fall a lot of mutations, but there are also mutations that are falling on the other positions. And these mutations, they have been linked to different diseases, like it was done in this paper. So in the lab, what we are trying to do, so our lab first is entitled Structure, Mechanism, and RNA Therapeutics. And in the lab, we are trying to determine the structural basis for RNA splicing regulation in order to develop novel therapeutic strategies and tools for oncology and genetic disease. So the discovery of small molecule splicing modifiers starts in the context of spinal muscular atrophy. So it's a neuromuscular genetic disease that causes muscle atrophy and weakness. And this is due to the decreased production of a survival of motor neuron protein that is called also SMN. And that triggers the degeneration of alpha motor neuron, muscle wasting until paralysis and death. So this disease is due to the gene SMN. So there is a copy SMN1, a gene called SMN1, sorry, which is the main source of this essential protein SMN. So it will be spliced in a way that the exon 7 will be mostly included, and this will lead to the production of functional SMN protein. So it's an essential protein. And these patients that suffer from SMA, they have lost their SMN1 gene, so they are expected to die. But in our genome, we have a paralog gene that is called SMN2 that differs from SMN1 only by silent mutation. And notably, there is a mutation here at the beginning of exon 7 that transforms a C into a U and will change the pattern of splicing of the messenger RNA. In most of the cases here, the exon 7 will be skipped, and this will lead to the production of an unstable SMN protein. So something which is important for this exon 7 is that it has a really weak 5' splice site. So first of all, the sequence of the 5' splice site is not perfectly complementary to the U1 snRNA, since there is an A in position -1. Then second, it's sequestered into an inhibitory secondary structure. And third point, it's flanked by a strong repressor that is called ISSN1 and will recruit HNRNP-A1. And so the strategy developed at the lab of Adrian Kreiner was to use an antisense oligonucleotide in order to mask this strong repressor and therefore boost the exon 7 inclusion and to produce functional SMN protein from the SMN2 gene. So this strategy works and was also validated by FDA and it's used in clinics, but it's an antisense oligonucleotide, and therefore it's difficult for it to cross the blood-brain barrier, and it needs to be injected in a repetitive manner directly in the spinal cord of these child. Therefore, the pharmaceutical company, they have looked for orally available alternatives that trigger the same effect. And it's only in 2014, so 10 years ago, that the SMNC series of small molecule was published in Science. And these small molecules, they can basically correct the splicing of SMN2 exon 7 in a dose-dependent manner. They also promote the production of functional SMN protein from the SMN2 gene. Therefore, they have the same effect than the antisense oligonucleotide, and they were qualified at the time as the splicing magic bullet. However, at this time, no one knew how it worked. And so what we've done, we started a collaboration with Roche, and they gave us the small molecule, and we tried to first to understand what's the specificity of this small molecule in a genome-wide manner. And when we treat cells with this small molecule, we observe that only a small subset of exons will change their splicing upon treatment. And when we analyze the 5' splice site of these exons, what we've seen is that there is an enrichment compared to the genome-wide 5' splice site consensus here at position -2 and -1 for a GA motif, and therefore it's a GA-GU motif. So it was also shown in another study from Novartis that we also identify some small molecule splicing modifier for the SMN2 exon 7, that the effect goes through the 5' splice site, because when they introduce this part of exon 7, SMN2 exon 7 into another genomic context, or in the case of BRCA1, they observe that this exon 18 became sensitive to the small molecule. So what we did first, we tried to bind this small molecule directly on the 5' splice site, and it was not working. And then we decided to test whether this small molecule could be able to bind the duplex formed by the U1 snRNA and the 5' splice site. So we prepare this RNA, and then we titrate it with the small molecule, and we follow the NMR spectra of the RNA. And what we observe is that there are changes that occur. And these changes, they do not occur when the small molecule is slightly different and who has also an activity which is strongly reduced. It means that this interaction is specific, and using sparse NMR data, we could propose that the small molecule is binding here the major groove close to the GAGU motif on the opposite face that is bound by the U1C zinc finger. So the zinc finger will just bind the back, the minor groove of this RNA helix and will stabilize the duplex. So we continue this work, and then in the lab, we also develop an approach to reconstitute in vitro U1 snRNP and associate it to different splice sites. We validate this reconstitution using a small angle neutron scattering, cryo-EM, NMR spectroscopy, and we could also show that it's able to bind messenger RNA containing a 5' splice site. And therefore, we also test the specificity of the small molecule in the context of the reconstituted U1 SNRP that contain either the 5' splice site of SMN2 exon 7 or a canonical 5' splice site. And we did that using NMR spectroscopy, using the fluorine atom that is present on the molecule SMNC5. So we put SMNC5 in solution, and then we add our complex U1 SNRP with the 5' splice site of SMN2 exon 7. And what we observe is that the fluorine signal is moving and then suddenly it disappears. So meaning that there is a direct binding between the small molecule and our RNP. If we add some RNAs, the signal came back to the same position. And if we do the same titration with a canonical 5' splice site, we did not observe similar changes. This signifies that in vitro, SMNC5 is selective for U1 SNRP bound to the 5' splice site of SMN2 exon 7, and this interaction is RNA dependent. We went on and solved the structure of the RNA duplex formed by the U1 snRNA 5' end and the 5' splice site and the small molecule. And as proposed initially, this small molecule is crossing the entire major groove close to the GAGU motif, so close to the exon neutron junction, on the opposite face that will be bound by the U1C zinc finger. If we have a closer look at this interface, we see that this small molecule is composed of 3 cycles. So the most important piece is the central ring here that will insert between the 2 cytosines and then will contact directly the unpaired adenine in position -1. The third cycle here, which is positively charged at neutral pH, directly interacts with the backbone of the U1 snRNA. This means that this small molecule is the minimal splicing factor because it will create a link between the U1 snRNA and the pre-mRNA. This interaction is specific since if we change this A-1 by a C, a U, we still observe a small residual interaction, but when there is a G, we close the bulge, so basically this G will base pair with this C, and the small molecule does not bind anymore. So we had a model that say that basically this small molecule will bind in the major groove and will stabilize the unpaired adenine in position -1 in the RNA base stack. And in order to validate this model, what we've done, we transfect X cells with different mini-genes containing the SMN2 exon 7, but they differ at position -1. There is either A, a G, a C, or a U. And then we test the activity of the small molecule at different concentration. And what we observe is that the optimal activity is obtained when there is an A in position -1. When there is a G, there is always inclusion of the exon. When there is a C, there is still alternative splicing, but it's totally insensitive to the small molecule. And when there is a U, there is still alternative splicing. And this, there will be an effect, but at really higher doses. So the bulge adenine in position -1 is essential for optimal activity of SMNC5 in cellular model in agreement with our structures. So based on this work, we proposed the following concept that we called 5' splice site bulge repair. So when the 5' splice site is canonical, the G is base pair and it's embedded into the RNA base stack. When there is an A, this A will be unpaired and will be able to oscillate between inside and outside the RNA base stack. And this will cause some clashes with the U1C zinc finger in the minor groove. However, when the small molecule is present, it will interact with A-1 and it will stabilize it into the RNA base stack, and this will transform this weak 5' splice site into a stronger one. So this was the elucidation of the mode of action, but the problem was that SMNC5 indeed did not pass the clinical trials because it was metabolized by the cells. And therefore, during this time, Roche tried to identify some other molecules that have still the same activity, but won't be metabolized into the cells. And so they identify, of course, the well-known risdiplam that has been later FDA approved, but they also identify SMNCX, SMNCY, and the molecule from Novartis that I talked at the beginning was also repurposed in another context, in the context of the Huntington disease, where it will basically favor the inclusion of a poison exon in the mRNA encoding for the mutated Huntington, and this will trigger its decay. So basically, we took this group of 4 other small molecule splicing modifier, and we tested whether they interact with the RNA duplex in a similar manner. So we grouped them into 3 different chemotypes. So there is risdiplam and SMNCX that have the same chemotype than SMNC5. Then there is the chemotype number 2 that contain only SMNCY. And then there is the chemotype number 3 called branaplam. So we tested the interaction using NMR spectroscopy with the same RNA duplex, and we observed very similar changes in the spectra of the RNA upon addition of the small molecule. So there is always this G+1 immunoproton that will be destabilized upon addition of the small molecule. And then there are these two pyrimidines that are shifting all the time upon addition of the small molecule. And this signifies that basically they will bind the same RNA binding pocket. And in order to find out the detail of this interaction, we decided to determine the NMR structure of these different complexes. So first, let's make a stop on the chemotype number 2. When it was initially published by Roche and in this publication from Eric Pinard, you see that they identify already that having a fluorine here at this position was very important for the activity of the small molecule. And us, using NMR spectroscopy, we could show that indeed there is a pseudo H bond between this fluorine and this amide proton. So we could show this using first 15NHSQC that identified the proton that is attached to the nitrogen, so it's this one. And then we could also show the proximity between the proton and the fluorine using a correlation through the space. So this means that by doing this pseudo-hydrogen bond, basically this entire central entity became flat. And this will be important for the structure later. In the context of branaplam, it's a bit different. So there is a tautomeric exchange between these two forms, where there will be either an alcohol function here or a carbonyl group here. And in order to determine which tautomer form is the one found in solution, so we perform NMR experiment, and then we look at the carbon chemical shift of this quaternary carbon here. And we could identify in our condition that it's only the alcohol form that is present in our solution. And therefore, we use this tautomeric form in order to solve the structure of the complex protein RNA. And here are the 4 additional interfaces that we identify. So basically, we identify that risdiplam binds very similarly than SMNC5. So it will place its central aromatic ring between the 2 cytosines here and direct its carbonyl in a way that it can interact directly with the unpaired adenine -1. In SMNCX, the carbonyl will be replaced for its role by this nitrogen here. And for SMNCY and branaplam, this planar entity that are formed here through the weak interaction will insert again between these 2 cytosines and lock the position of the oxygen in a way that it can accept hydrogen bond from the unpaired adenine -1. So to summarize, our work explains why how these small molecule splicing modifier are working. So there are molecular glues between U1 SNRP and given 5' splice sites. They stabilize the bulge adenine in position -1, and they are allosteric activator of the SMN2 splicing reaction. And based on this data, we derived the mechanism of 5' splice site bulge repair. And the second part of this work show that the diversity of splicing modifier acting on A-1 bulge 5' splice site reveals some rules for the rational drug design. So it means that these 5 molecules, they are interacting similarly with this RNA duplex, and this will give rise to similar activities. So it's still a bit puzzling for us what this part of the molecule is doing, but the central ring will definitely lock the adenine -1 in the RNA base stack and fit and stack, between these two cytosines, while this part of the small molecule, which is positively charged at neutral pH, will form a link with the U1 snRNA. So there are still some open questions in the field regarding A-1 small molecule splicing modifier, but regarding also small molecule splicing modifier in general. And this is due to the recent publication of new data from the lab of Carine and Tisné in Cold Spring Harbor. So what they did, they used risdiplam and branaplam, and they perform, basically splicing assays in the context of the SMN2 mini-gene using a lot of 5' splice site variants. And then they use sequencing to quantify precisely the dose response curve of this small molecule. And in fact, what they found that risdiplam has a consensus, a preferred consensus that contains the GAG motif, as I mentioned before. And risdiplam will correct the splicing on all the exons that contain this sequence, while branaplam will also correct the splicing on this type of 5' splice site, but also on this type of 5' splice site that are enriched for A in position -3 here. So you have few examples of genes that contain these different consensus. So the SMN2 contain the GAG motif with A in position -3, and while the HCT, with A in position -4, yeah, like the FOXM1, and so these are two major targets for risdiplam, and while HTT, so the Huntington that I talked about before, and also SF3B3, they contain an A in position -3. So it will be very interesting to determine if there are contacts between branaplam and this A in position -3. Another important question in the field is, can we change the specificity for the base in position -1? Until now, all the SMNC compounds, but also we have seen branaplam, they act on a GAG motif that contain an A in position -1. Would it be possible to identify some small molecule that will correct the splicing when there is a C or U in position -1? This remains an important question in the field. And finally, is it possible to change the position of the bulge target by the splicing correction? It seems that the answer is yes, according to recent data that have been published by PTC. So they looked at the cause for familial dystonia, and they look at the splicing change of the ELPN1 gene. So in the wild-type version, there is a T in position 6 that will lead to mainly to exon inclusion. So as you can see here, however, in the people suffering from this from the disease, this T is transformed into a C, and this will lead to mainly exon skipping. So I mentioned you before that the bulge repair will act through U1C, so at position -1, and here we are quite far from this position. And in the structure of the U1 SNRP from yeast, there is also another zinc finger that binds this part of the RNA duplex that is called LUX7, and that could basically be an analog for the U1C and explain why this small molecule identified by PTC, they can correct the splicing of this exon. which has a mutation at position -6. It will be very interesting to understand how these small molecules are working. So I'm done now with my talk. I would just like to thank the people that have been involved in this work. So this work was done primarily at ETH Zurich under the supervision of Professor Frédéric Alain, in collaboration with Roche, but also with Skyhawk Therapeutics. And now I'm doing this job at the University of Bordeaux in the RNA unit, where I'm hosted by the European Institute of Chemistry and Biology. And I would like to thank in this context, particularly Florian, Julien, Léa, Agathe, Julia, Cameron, and Sébastien. Thank you for your attention.
Thank you very much, Sébastien. So if you have any question, please write them in the Q&R. In the meantime, I just want you to stay connected because I have two announcements by the end of the question session. So please remain connected. So the first question is to know if the main determinants for binding of those drugs are at position -1 or -3. And so how can they have this so specific effect on splicing regulation?
So the main position that is targeted by this splicing modifier is the position -1, and it should be in the context of GU.
Okay, and so it's very specific because you only have at position -1. It's sufficient to be so specific?
No, there is also something that I did not mention because it was under debate and it's still under debate, is the exon content. And in the exons that are sensitive to the SMNC compounds, there is often a polypyrimidine tract, polypurine tract in the exon. And this part of the exon is important. And what we could show in the past is that this part will recruit some splicing factor that will help also the recruitment of U1 SNP. And so the addition of the natural splicing factor and the artificial splicing factor will both participate, to the recruitment of U1 SNP on these 5' splice sites.
Okay. One more question is to know whether if A-1 is methylated at position 6 to form M6A, does it affect the splicing efficiency?
I have no clue.
No clue. Okay. I may have one question. So you mentioned the RNA-seq data from Carine's lab. So did they observe many RNAs for which the splicing is affected by the incubation with those two drugs? Or?
No, they only observed the 5' splice site. So it was a focused screen where they just changed the 5' splice site. Okay, and they observed, like it was shown before, that it's dependent on the GA-GU motif, and they found out that for branaplam, the position -3 seems also to be important.
Okay. But they never did some experiment in human cell where they treat the cells with those molecules and they look at the splicing.
So we did that already
by RNA-seq.
We did that already before in 2017, and we have seen that there is only 20 to 25 genes that are strongly changed upon addition of this small molecule.
Okay, impressive. Any more questions? No, so maybe let me announce two things, and then maybe there will be more questions in the Q&R window. So the first announcement, is to let you know that the next RNA collaborative seminar will be hosted by the RNA Research Center from Poznan in Poland, and it will be on September 25th from 4 p.m., so same time as today. And the second one is to let you know that the 13th CIFRN meeting, so which